So if you look at the history of mathematics, it has in many cases learned a lot from plant science. If you think about the beautiful Fibonacci patterns that appear in the positioning of leaves, or the phylotactic patterns that show up in sunflowers, or even the wonderful coiling of the tendrils of climbing vines that people like Darwin and others have looked at, that's been a source of motivation for a lot of very, very interesting mathematics over the years. And today's lecturer, Jacques Dumais, is one of the great practitioners of this science at the intersection of mathematics and plant science, actually. And like many, but not most, but many distinguished applied math types, he actually began life doing something completely different. Namely, he got a PhD first in biology from Stanford University. He then came to do a postdoc, actually, in our Department of Applied Mathematics with Mahadevan in the early 2000s. He moved to Harvard University as a junior faculty member after that, but the lure of South America was too strong, and in 2012 he moved to Viña del Mar in Chile at the University Adolfo Ibáñez, pardon my Spanish. Jacques has done an incredible amount of interesting work on mechanics in plant science, including such wonderful problems as the way the Venus flytrap traps. And today he's going to tell us about some geometric problems that have been motivated by plant science. Jacques.
Thank you, Ray. Can you hear me well? Yes? It's really a great honor to be here. First and foremost, because I'm a biologist. So I'm not, even if Ray would like to say that I'm an applied mathematician, I'm really a biologist. That's my background. But I do have the greatest respect for mathematicians and for the work of mathematics. And so what I want to tell you is a bit of a journey that we've made, or that I've made personally, in trying to find footage, or find areas in biology, in plant biology more specifically, where there is some mathematics, worthwhile mathematics to be done. So I will even tell you a little bit of my personal history. When I was a student, an undergrad in studying biology, I was looking for some area of biology where you could find what I would call theoretical biology, where you could do something that is quite formal and perhaps have theorems or have something quite precise and still be relevant to biology. And there was, maybe some of you will know, there was a movement in the US in the 30s and 40s called mathematical biology or theoretical biology, led by Nicholas Rashevsky, where they were indeed trying to find an axiomatic, or I have the foundation of an axiomatic biology. Very much in the same way that you have a theoretical physics, they were trying to have a proper theoretical biology. So what do I mean by that? So, and the question is really, does it exist? Can you do theoretical biology? And I think most of you will agree, there is a lot of theories and models in biology. Is there a pointer here? Yeah. So there are certainly a lot of mathematics done in biology, but the typical path to a model is one of induction, if you will. You would start with some observations or experiments, and then someone will try to make a theory or some models based on these observations. But what I'm looking for is really have a basis, a mathematical basis strong enough here at this level to be able to infer and predict things that are new about biology. And that's the challenge. And physics has achieved that. I think there's a theoretical physics where you can make sensible prediction about the physical world before having observed it or experimented it. The question is, does it exist in biology? And so that was what I was looking for as a young scientist. And Ray touched on one aspect of plant biology where there is something quite formal, and it's phyllotaxis. And I won't talk about phyllotaxis today, but you probably know this. This is sunflower. If you were to count the spirals in the head of the sunflower, you would find that most of the time you get a number from the Fibonacci sequence. And in fact, by the time you reach a sunflower, it's probably something like 55, 89, the number of spirals you find there. And there are exceptions, and even the exceptions tend to be, can be explained with a slightly more general model. And there is a paper by Stéphane Douady and Yves Couder, who's sitting right there, that really made a big impression on me. So it's a PRL paper that was published in '92, where they have a simple dynamical system. And this is just the bifurcation diagram of their dynamical system. And they show that this main sequence here, the Fibonacci sequence, and therefore the patterns you observe in plants, are in fact the main branch of their dynamical system. And so this was, I felt, a very nice way of starting to formalize biology. It's not all of biology, obviously, but having a proper theoretical biology. 10 years later, if I put this here, I met three true mathematicians, so Pau Attala, Christophe Gollet, and Scott Houghton, who were also working on phyllotaxis, and formalize, if you will, the dynamical system that Douady and Couder had put forward. And in their paper, they come up with this, they explain this bifurcation diagrams that we have here. But on top of that, they were looking for periodic orbits, and they found one here, a periodic orbit, which is looking at the angle between the organs when a structure here, this is another plant structure when it's growing. And they show that this periodic sequence, in fact, reproduces patterns that biologists had observed, so quite closely. So they have 130, 89, 89, 130, 89, 89, 130, 315. That's their periodic orbit. And this biology, Shirley Tucker, had observed in some of these plants here this sequence, 135, 90, 90, 135, 90, 90, 135, 315. Okay? So it's the kind of information, if you're just a biologist and you see that, well, you say, it's life. It's complicated. Can't explain it. But it turns out there are simple models, mathematical models, that do explain what might seem on the surface like a very, very complex process. So that was, if you will, how I started in this area. And it's, again, a few years later, I stumbled on this problem, which is a plant. In my case, all the problems, they come in form of a plant. And so what you're looking at here is a gland. So this entire structure is a gland, and those are cells. So there is some symmetry here. It looks as if it's not a completely chaotic system, but it's not such a subtle, it's a fairly complex structure. And what I wanted to do is see if we could explain these patterns. And biologists have known for a long time that there are rules when a cell divides. So the idea is to see if we can, based on some basic rules for cell division, can we explain these structures here. So it's old rules that were put forward by one German botanist, Julius Sachs, who observed a number of plants or cells when they divide, and he inferred two rules from that. One of them was that when a cell divides, it will create two daughter cells of equal sizes, which seems like a decent observation. And then he also observes that new walls, when they meet an older wall, so I should say the plant cells is surrounded by a wall. So when I'm talking about cell wall, I'm referring to the edge, so the contour of that cell. So the new walls that are laid down by a dividing cell will meet the older wall at 90 degrees. So they're orthogonal to the old walls. And finally, there is another rule that came out a few years later by Leo Herrera, who was in fact working side by side with Plateau, and he made a parallel between cell division. He said that the plane of division behaves as would a soap film placed under the same condition. Okay, so Plateau had observed the law for the equilibrium of soap films, and Herrera realized that some plants, or in fact many plant cells, when they divide, seem to be approximating what a soap bubble would do. And the idea is, as you probably all know, a soap film will try to minimize its surface area. So by making this parallel between soap films and plant cells, Herrera was saying that a dividing cell will probably look for a division plane that minimizes its area. So that's the premise that I want to use, rules that are empirical rules at this stage, and see if we can formalize these rules and predict some patterns that are observed in plants. And there is an old problem that's related to that, a mathematical problem this time. In fact, that was worked on by Norbert Wiener here in Cambridge in a very short paper that he called the shortest line dividing in area in a given ratio. Wiener raises this question here. He says, let us suppose a farmer wants to divide an irregular field of his evenly between his two sons. Now we'll probably say his two children. And suppose that he wants to use as short an edge as possible. What shall he shape the edges? His fence. What shall be the shape of this fence? So that's the idea. We've got, imagine any shape that you want. We'll probably assume a convex shape for the time being. And I would like to divide it in two equal areas using the edge, or in my case, a cell. So it's a cell wall that is the shortest possible. And so in this paper, Wiener works out the solution for circles. So if your field is a circle, how would you divide it? And he finishes his paper saying it is almost self-evident that the shortest line to divide a convex area in a given ratio is a single arc of a circle, but this I have not been able to prove yet. So it is, I'm sure you agree, almost self-evident. For some reason, Norbert Wiener decided not to use the calculus of variation to make his proof in this paper. So he's using, in fact, geometrical constructions. He doesn't want to use the calculus of variation because he's concerned that some of the assumptions of this problem here would not be easy to formalize or to state using the calculus of variation. We actually used the calculus of variation to find, at least under some conditions, whether this statement that an arc of a circle would be the shortest path possible, to find if that was correct. So we framed the problem this way. We have a cell. So this is what I call the wall or the edges here, and I want to divide it with whatever function that would be the shortest possible, such that I have two areas of equal size. So we have this length here, and we want to minimize the length, find the function that minimizes the length such that the area is half of the area of the mother cell. And so you can solve this problem. And if you do so, you find that the function must satisfy this condition here that you'll recognize as being the curvature. That's the Lagrange multiplier. So the curvature is equal to a constant. And what you also get from this solution, you get this relation for the derivative of the dividing wall and the edge of the cell. So you dy dx, df dx. At this position, x1, and also at this position, x2, must be equal to -1. Okay? So that comes for free, if you will, when you minimize this system. So it's telling you that you need to have walls that are meeting at 90 degrees over here and here. Okay? So, in fact, it looks as if there are two rules, and the third rule is a consequence of the two rules. There is Era's rule that wants to minimize, that tells us that we want to minimize the area or the length of the dividing wall. And then there is this first rule by Jacques that tells us that there is an area constraint. And by solving this problem, we also find at the same time that the walls ought to meet at 9 degrees with the outer perimeter of the cell. So we decided to see if there are some predictive power in these rules, and we followed some work that was initiated by Darcy Thompson. Darcy Thompson did use you probably know of his book on growth and form, so 1,000 pages of applications of mathematics to biology. He probably has at least 1/10 of that focused on cell division. And within these 2 or 3 chapters that he has, he discusses a lot of cell patterns that are like this one. So this is a gland, but the same gland that I showed you earlier, but a bit at early stage. And so you'll see it started its life as essentially a flat two-dimensional disk. That was my initial mother cell. And then it went on through several rounds of division and created this pattern. And in his book, Thompson shows, well, first of all, if you do have a circular cell, it's pretty obvious that the diameter is the best way to divide it. So the shortest possible way to divide that cell was with this diameter here. And then the next division is also trivial, so you would expect that the other transverse diameter is the best way of dividing these daughter cells. But then you're left with these, what we call quadrant cells, and it's not so obvious in this case. How would you divide it? And certainly if we were to keep doing the same thing, cutting along radii, I would cut it like a piece of pie here. This, if I were to cut like this, it turns out this edge would be longer than the one we observe here. And so Thompson shows, it's a simple calculation, that indeed a division like this one is the shortest possible way of dividing this quadrangle here into two different cells of the same area. So and, there are a few more examples to convince you. I'm a biologist, so I need to do experiments to convince me that whatever calculation I did was correct. So here's my experiment. This is a Petri dish. You're looking at a Petri dish with two plastic walls here that mimic essentially the first two rounds of division. And then we put two soap bubbles in each of these quadrangle here. And you see how they find their equilibrium, which is exactly what the plant is doing here. Okay? So that was known for almost 100 years. Darcy Thompson published his book in 1917, and therefore it's been around for a long time. And by and large, it was forgotten or left on the side because biology was moving on into molecular biology, a lot more precise in terms of what is going on when a cell divides. And therefore, all of these more geometrical models fell by the wayside. But I got excited about this because we started finding exceptions in how cells divide. And so here, all of the green cells you see here are exceptions. So this is one quadrant cell that divided this way. So this I'll call periclinal. This is called anticlinal. So this is the shortest, this division here would be the shortest possible plane for a quadrangle cell. But there are some cells that will divide like this. And here's another one here, and so on. So there are exceptions. And we got excited about this when we realized that, in fact, these exceptions are also predicted by soap bubbles. And here is, again, like an experiment. I need to do an experiment to convince myself that what I'm seeing or what I'm calculating makes sense. So we return to this Petri dish, and we have two soap bubbles. And so it is possible to have two soap bubbles in equilibrium in this configuration. So just like in this configuration. But look what happens if you start playing with, where is my cursor? Here it is. So the good thing about soap bubbles, you can play with them. It's much harder when you're doing biology. So we have a little needle here, and we're moving the soap film to the edge, and we can have it switch back to its other configuration. Okay? So there are indeed two equilibrium configuration, or a third one, if you will, if it would be the mirror image of this one, so you could have a division like this here. So there are three configurations that are possible given two soap bubbles constrained by this outer geometry. And you can, in fact, with soap bubbles, you can in fact measure, and when we did this movement here, we can do it precisely and measure the work that's being done to go from one configuration to the other one. And this is the energy landscape as a function of position. So this was the solution that we found earlier with this periclinal division. And then if you move this edge, you can move it either by pulling on it upward and measure the force. You find cusp. So there's a cusp here, and then you fall into another minimum. So it is true that the global minimum is this configuration, but there's also a local minimum that corresponds to this geometry. So we started asking, what exactly is a cell to do Why does a cell might fall in this minimum versus this one? Why is it that some cells divide like this? It's a small fraction. About 5% of the cells will divide like this. Most of them will divide in this configuration. So we started looking at a different system, Coleochaete, which Ray asked me to put on the slide here because he loves Coleochaete and all plants, everything, all the algae. Let me tell you, I'm going to be talking quite a bit about Coleochaete, so let me tell you how it grows. It's a two-dimensional tissue. So what you're seeing here, this is a cell, for example, this is another cell. But it only grows at the perimeter here. So you could imagine it's a front. You have a line of cells on the perimeter here. They all move outward. And as they grow, when they reach a critical size, they divide. And they divide according to their geometry. So Coleochaete is, because of its simple growth and geometry, you can do some precise characterization of cell shape. So if you were to focus on the edge here and look at the geometry of the cell, that's what you would see. There are so the cells are trapeze, if you will. They have two outer lateral edges that correspond to radii in the colony here, and then the inner edge and the outer edge are arcs of circles to first order. And so a geometry like this one you can characterize with a two-dimensional shape space. You can use the angle between the two lateral walls, so these two walls here, and you can also use the ratio between the height of the cell and its radial position, okay, the radius of the colony. And so we normalize it. Its size in this case doesn't matter. We just want to know what is the shape of the cell. So here's the space, the angle here, and the aspect ratio of the cell. And we've subdivided it in terms of how the cell would divide were it to fall in this region or this region or this region. So the blue region corresponds to cells that are fairly wide, and therefore they divide along this edge. The red region are cells that are fairly long, and they divide using this edge here. And in this little wedge, you have cells that are such that this solution is the best solution. Okay? So these lines are just, you can calculate when you go from one type of solution to the other one. And so what we did, we went around and measured thousands or hundreds of cells here when they're just about to divide or right after division, and see how they were dividing. And this is what we found. So every point here is a cell that we measured, and its position in this space, it corresponds to the shape of the cell that we observed. And then we colored every point based on how the cell divided. So every time a cell is blue and sitting in a space that's blue, we're fine. The model works. A red on red is good. Green on green, you probably can't see, but there are a few green cells here. Green on green is good. And by and large, you can predict. If you give me the shape of the cell, I can map them into this space here and tell you, well, it stands here, it sits here, therefore it will divide according to this plane of division. But there is noise, and we were intrigued, in fact, by the noise. And if you look at carefully here, you'll see that there is more noise in this region. So when you move away from this line here, you're getting quite solid blue, except for an exception. We're allowed, it's just biology. And same thing here. If you look on this side, you have some exceptions near the border, and then it becomes pure red. And I'm sure you understand that what this border represents. It's essentially a cell geometry, where there are two division planes that are equal. And therefore, the cell, you're asking the cell to distinguish between planes of division that are quite similar, nearly the same length. And we can show that, in fact, if you project, you could imagine drawing a line here and projecting all the data onto this line and calculating the probability of dividing according to the blue division plane or the red division plane. And this is what you find. So 0 here corresponds, the delta is just a difference. Delta 1, 2 is a difference in the two different solutions, the length of the two different solutions. So 0 corresponds to this boundary here in shape space. And then positive value, 0.5, for example, would mean you're deeper into the blue space. Minus 0.5, you're deeper into the red space. And we've mapped here just a proportion of cells that are dividing according to the red plane or the blue plane. And what you find, if you're far enough in the blue space, you're only dividing according to the blue plane. If you're far enough in the red space, you're only dividing according to the red plane. And then there is a transition region in between where the planes are close enough, they're not zero or close enough, that the cell has a hard time telling the difference, and therefore will sometimes fall in one plane or the other plane. There are many more examples. The summary is here. What we found is, for other cell geometries, is always the same trend. If I'm going to use a rectangular cell as an example, if the cell is a square, the two solutions are exactly the same. What you find is a 50-50 split between these solutions. So this would be, again, the difference in length of the two different solutions. That's the frequency. So obviously, a square cell could not tell which one is shortest, and therefore would choose either side with equal probability. But if you start stretching the cell, by the time you reach an aspect ratio of about 0.2 or a length difference of 0.2, you get almost 100% precision. So it's as if the cell has some ability to tell the length of respective or competing solutions, but does it with some error. So it's only when you reach somewhat skewed cells, a 20% or so difference, that the cell can reliably decide and find the shortest division plane. And this is all published, so I'll just mention quickly. We've looked at a number of systems here, and we also have an equation. This is in fact a fit using the Gibbs measure. We can show that there is a molecular mechanism, or simple molecular mechanism, that explains how you get from a 50-50 split between different solutions to a perfectly deterministic division. So I'll pass this because it's been published for a few years now. But I want to focus now, in fact, in cases where the difference is fairly large. So I'd like to look at cells that are, or at least make that hypothesis, that the cell is a very good divider, will always find the shortest possible division plane. And I wanted to do that because I'd like to work on a precise deterministic dynamical system. So the idea is simple. I'll look at a shape space. So we're still using 2 dimensions to keep it simple. This space describes the shape of the cells that I'm looking at. And I'll define 2 different maps or functions on this space. One of them, so imagine I'm starting at time t. I have a cell sitting here. Before I can divide it, I'll need to grow it. So there's a first map, G, that grows my cell. And depending how we choose the map, this map could change the shape of the cell. And therefore, you might map it somewhere else in this space. And then I have a second map, which divides the cell now in 2 daughter cells. Okay? And now I would have to choose. And the choice is often easy for us when we do simulations. We track one type of cells over time. But so there is a little bit of fuzziness there because we have to choose one of the daughter cells to know where we stand. But if you choose one, so you'll find the new location of this cell. So this is S of t plus 1. So this is the simple dynamical system that we want to look at. And we would like to apply it to Coleochaete and other systems to see if we can make some predictions. When we started showing this data, I'm back to Coleochaete, a lot of people mentioned, why is it that you're not using all the space? And there is, in fact, a very well-defined cone shape here. So what is this exactly? Why are the cells confined to that wedge? And so having raised the question and having a dynamical system to study how cells are evolving, we started looking at that. So you could imagine a cell, in this case, sitting in the blue plane. This cell, when it divides, so here's my cell here. When it divides, what happens is it cuts the angle in half, but doesn't change the ratio h over r. So this cell, when it divides, it just moves halfway here between where it is and the origin. Okay? It just divides the angle in half. So it's a sit here. And then this cell is not, so this is just the process of division. It's not ready to divide again. It needs to grow. And to grow in Coleochaete, I told you, you grow only radially. So this cell has to grow stretching upward here. And the process of growth will not change. So here's the growth process. It doesn't change the angle. It just changed the ratio of the height of the cell over the radius. Okay? So you've essentially, by dividing, you're moving, you're cutting the angle in half. So you're moving halfway towards the y-axis. And by growing, you're moving upward when you're sitting here. So you could imagine the image or doing a map of the entire space and see how it lands, where it lands in this red space. And it's enough, in fact, to look at this edge here and map, so the boundary between the two regions, and see where it maps in this space. And this is what you find. So you find that points that are sitting on the blue side, quite close to this line, will map into a line. So it's the image of this line here, of this curve, into the red space, and vice versa. You could start with a red cell and divide it and grow it, and you would find that it maps this line here into the blue space right here. Okay? So the dynamics of the division essentially forces you to move as far. The farthest you can move is to this line here when you're on the blue side, when you begin on the blue side, and if you begin on the red side, you move down to this line here. And if you look at the data, it fits perfectly. So you see the curve here. It predicts perfectly where these cells are standing. Okay? So we were quite pleased by that, to see that we could not only predict how cells divide, but also predict the kind of evolution that they'll have. Where a cell, once, if it's born here, for example, where is it going to move after some time? And it's constrained by these boundaries, so it's going to be bouncing back and forth between these two spaces here. And what we showed is, in fact, that it's moving down an attractor. So I can summarize here, just to repeat briefly. A blue division makes you move this way. So you divide first the cell and then you grow. So that's the typical path that you're doing. So you're going to the left and upward. And a red division gives you, in fact, forces the cell to come down quite a bit. And then as it grows, it will come up slightly. If you were to sum up these two processes, you essentially have one process here that forces you to the left. So whenever a cell happens to land in the blue region, it will have its angle, theta, divided in half, and therefore there is always a constant flow towards the left. And then comparing the movement associated with the blue division plane and the red division plane, you can see also that there is a net flow downward. Because you go up when you divide with a blue plane, but you go much more down when you divide with a red plane. So the sum of these two trajectories gives you a downward flow at the same time. So we actually color-coded the points to show that these points, the white points, represent cells that were young, and therefore early in the colony, so it's a early time. And and the dark cells here represent cells later on in time. So after many iterations of this map of dividing and growing, you find that a cell that might have been born here will flow slowly down to this attractor. Okay? Now there is this attractor is interesting, nothing very exceptional. You can imagine what I don't have. Well, I can show you So we're talking about cells that are right here at the margin when the colony is quite large, cells that have divided many times. So here's the cell. So you see that they are almost like rectangles. And the map, so basically when you're here, the angle between the two lateral edges becomes 0. This ratio h over r also tends to 0 because now the radius is quite large. You're far away from the beginning of the growth. And so you only have this map here, essentially a cell that will grow to twice its length, and then it will divide to form again 2 squares, okay, and keep growing this way. So here it is. This is Coleochaete, and this is a simulation where we simply use these rules, these division rules. You can see that we mimicked almost perfectly this colony. Okay? And I need to emphasize, this is all done without parameters. We've just begun with we start with these 2 division rules. We formalize them, and and everything else is frozen. I can change the initial condition. I can make it run faster or slower, longer, but I cannot change the solutions. There is no free parameter that I can change to try to fit reality. And so in this sense, it's a model that's predictive without having this fudging factor or some parameters that you can vary just to try to fit what you observe. Okay, keeping with this idea of looking at this system as or these division as a dynamical system, I want to look again at the flow within shape space, but I'd like, in fact, to look at fixed points in a simple case where the growth process doesn't affect the geometry of the cell. So that would be a simple scaling. Once I have a cell geometry, I just grow it by scaling it by twice its size, and then I divide it. So division itself will change the geometry of the cell, but growth is just a scaling. It doesn't change the geometry. And so the fixed points would be defined like this. You want to look at the map of S of t, such that S of t plus 1 is equal to S of t. And one simple one would be this one here, square or rectangle cell. So here's the mother cell. It divides. So this is obviously the shortest way of dividing this rectangle here. And you find that the two daughter cells here are exactly a copy of their mother cell. And you could repeat this over and over, so you would get that sort of pattern. So we had a large rectangle, which we divided it, and then this daughter cell would divide again, and so on. And we always keep the same geometry. Okay? This you've seen before. This is the A paper format, which was probably invented for this reason. It's very easy to fold it, and you always keep this somewhat aesthetic or balanced aspect ratio for the piece of paper. But there are more. So this is a trivial one. There are some more interesting fixed points, and here's a list with a different number of sides. So this is the case we just looked at, this rectangle. This cell is nice to look at. So this is a five-sided cell. And you'll see that the daughter cell here is exactly like its mother. Here's a triangular cell. It's not equilateral. That's a scalene triangle. And this little triangle here is exactly like the mother. And so on. So there is a two-sided cell that you could think of, and also a one-sided cell, which is just a circle dividing like this. Okay? So this is what you would predict from the map, from the division rules. I'll come back. These, if you think about it carefully, they're not minimizing, this division plane here is not minimizing the length. So in a second, I'll tell you under what conditions you could find a division plane like this one. But certainly, all of these three cases, it's not only do we have self-similarity, we have a daughter cell that's exactly like the mother, but we do choose the division plane that's the shortest to divide this mother cell. So all of these are the shortest possible ways of dividing these cells. So we can show formally that these fixed points exist, or they're feasible. To find them, we'd have to show that they are attracting, that there is some sort of neighborhood around the shape that we look at that is attractive, where the flow leads to this fixed point. And so we've done it. We're in the process of doing it for a number of different geometries. But we've done it for in the plane for a triangular cell. So this is the attractor here. And as you know, a triangle can be defined, the shape of a triangle can be defined with just two angles. So the fact that they have curved edges doesn't matter. If you give me two angles, I can define the triangle. And so we started in this space here, triangular space, and we look at how, when you divide these cells, how they move, what kind of flow you get. And they all move to this point here, which is this fixed point. Okay, so there is these fixed points. Not only they exist, there are attractors, and therefore they should be observable in nature. So we started looking for them, and I have a list of pictures here where we compare some organism, mosses and ferns and things like that, and these fixed points that we found. So here's one example of this is a young leaf in a moss, and here's it's called the apical cell. So there's a central cell at the tip that's growing and dividing, and I've labeled here to help the eye the different division planes that this cell has undergone, and it fits nicely this two-sided fixed point. The triangular fixed point is in fact all over the place in the biology literature. This is a fern. They have this apical, three-sided apical cell, and you see quite nicely all the sequence of division that has undergone in the past, and it fits quite nicely with the fixed point that we predict. And even the rectangular cell, there are plants that are more square in their way of growing, and here it is. So this is the apical cell, and you see little packets of cells that are also that have maintained this geometry or relationship that you see here. Okay, so we've got 3 cases here, or 4 cases, where we observe that there are some relation between these fixed points and what we observe in nature. We wanted to know why is it that this plant adopts rectangular cells, or 4-sided cells. This one will adopt a 3-sided cell, and this one here, this moss, adopts a 2-sided cell. Now, what exactly is the conditions? And if you follow, you'll see that the geometry of the apex, on the growing point, is evolving. In this case, you have a very large cell or a cell that's its size. It's on the order of the radius of curvature of the organ, if you will. It makes this cell here makes a large part of the organ that you're looking at. This triangular cell is now a bit smaller. So this is, if you will, this is curved here. So this is the size of the organ, and the cell occupies, I don't know, 5% or something like that. And then it gets flatter and flatter. So this is another growing point, the apex, and now the cell is getting quite small compared to the entire field of cells or curvature of the organ. So we realized that there was probably something about the geometry of the space on which you're dividing and growing that affects where you converge, 4-sided cells to 3-sided cells. So to keep things simple, what we decided to do is simply look at 2-dimensional cells still, but on a cone. Okay, so the cone is our way of modeling a meristem that it's pointed, or that's very, very small and sharp. And if you take the cone to be a plane, then you're modeling a large and flat meristem. So we're still looking at 2-dimensional division. But they're projected, so the cells are dividing, if you will, and growing onto this conical space. So that's the cell, really, this contour here is a 4-sided cell living on this cone, and this is just a projection of that cell in the plane above it. And if you do that, and we haven't finished everything, you find this. You find that if you run simulations on a very sharp cone, very pointed cone, you quickly converge to this solution. So those are, if you were to look from above, you would find concentric circles. So those are the one-sided cells that I showed at the beginning. And Cara, again, this is for Ray. So Cara is here. There are many more examples, but Cara is probably the best of them. So here's the apical cell that divides. There are 3 examples here, but this one it looks best like this one. So this is a sort of conical cell that grows and divides, and it has left behind this sequence of what would look like circles, just like this structure here. If the cone now, so you get this solution for angles that are between 0 and 15 degrees, more or less, with some overlap. And then as the cone gets flatter, you transition to, this becomes unstable, and you get a 2-sided cell, just like in this solution. And as you move further up to 55, 56 degrees, you now find 3-sided cells that become dominant. And as you reach flatter surfaces, you find that the square now is the dominant geometry. So we're still looking at exactly what is the range of attraction for the cone angles for each of these different cells. But there is, again, without parameters, the only parameter is the angle of the cone that we ought to change to mimic the geometry of the plants. But without parameter, we can predict the type of cellular patterns you would see in these plants. And it turns out it's not a biology audience here, but this is what I'm drawing here is the evolution of land plants. Cara is the ancestor of all land plants, and mosses are one of the first diverging group. Ferns and Selaginella are later on, they're the first vascular plants. And so we're essentially mapping this transition from pointed tip to a flat surface is mapping or following the evolution of land plants. I just want to finish with 2 more slides to see another point. If this map that we're using is, you probably realize, it's a map of a space onto itself. The dividing cell, the map that we're using is just, you just expand the space, cut it in half, and then you're still sitting on the same space. So if the map is continuous, you should be able to have some vanishing point. So there's a point or fixed point in space where all the iterations of these maps always line up. And I've done it for some reason, unknown reason, using the map of South America. And in fact, this map is converging on my office, more or less. So this is what you do. So you can start, for example, with a map of the large map here of South America. I'll shrink it in half, and then just lay it at random on this first map. So that's my second map here. And then I'll step on this map just like I was standing on the first map and do the same process. I will shrink it in half and drop it again. And so what you find if you iterate this process, there's always one point, at least one point, where all the maps are lining up. And so we would expect, because we were doing the same thing, we had a growing apex where we expand the cell, divide it, expand it, divide it, that there'll be also a vanishing point in the same way that this map is giving us a vanishing point. And there's a paper where they have a simple construction for finding what is that vanishing point when you have simple iterative maps. And the trick that they use is essentially relate edges of equal edges or similar edges between 2 different iterations of the map. So in this case, for example, this is the mother cell here. I would associate this point with the equivalent point on the daughter cell, and I draw a line between them. And I can do that for a few cycles, and the intersection of these lines gives me the vanishing point, okay? The point where the map is. And you see it, right? If I were to keep dividing this rectangular cell, I would eventually converge to a point around here. And that's indeed what we see. So this is another example of a 4-sided cell in juniper in this case. And you do see, so if I take a large domain that has time to grow, and I draw these diagonals here, I find that they converge also on this apical cell. And in the biological context here would mean that this is the center of growth of the meristem. This is where all the expansion of the cell is going on, or that's the central point, if you will, of the expansion map. And therefore, this is where the vanishing point has to lie. Okay, so I forgot to mention at the beginning with faces, the people who did this work. So I'm just going to step back here. So the work started many years ago, 7 years ago. And it was led by Sébastien Besson. So a lot of the early work that I described was done by a postdoc, Sébastien Besson. And some of the dynamics of that work was fine-tuned or developed by Antoine Lissé, who spent just a few months in my lab, but within these few months was able to analyze a lot of the fixed points of these cells. And more recently, Étienne Couturier, who is a true mathematician, so he's probably the right person to do for this job, started looking at the attractor. So how you can, or rather the basin of attraction of all of these different cells. So with that, I'll stop and I'll take your questions.
Thank you, Jacques, very much for that. because we're live streaming elsewhere in the globe at the moment, those of you who want to ask questions, we ask you to use microphones. And there are various microphones, I believe, around with some of the assistants, I hope. If not, here's one. So if you're anxious to ask a question, please identify yourself to one of them so that we can get you a mic. Questions? Here, let me.
I'd like to go back to right to the beginning of your talk, when you're comparing the development, the evolution of shapes, cell shapes with bubbles. So I'm sort of asked the question, if one sort of seriously studied the chemistry of a soap bubble, does that give you any indication of the chemistries that might be driving the cell configurations?
It's the gist of the paper I listed. So we have a PNAS paper that describes how the biology could give you at the macroscopic level something that looks like a soap bubble. And so I want to emphasize and make clear that there is no surface tension. When a cell divides and it lays down the new membrane and cell wall, there is no surface tension. And in fact, there is not even a plane that exists as such that could be minimized. So there is nothing there. So the model we have is in fact based on the dynamics of microtubules. So microtubules are involved in, well, you probably know the spindle that's involved in dividing the cells. In plant cells, before you see the spindle, cortical microtubules they're called, they live on the surface of the cell, will self-organize and start forming what is called a pre-prophase band, a band of microtubules that cinch the cell surface. And so we show that an interaction between this band on the surface of the cell and they call endoplasmic microtubules, so microtubules within the cytoplasm, could lead to this type of behavior. So it's completely, the physics has nothing to do. It's a self-organization process. It's not an energy minimization process in the plant. So there is energy involved in getting to that plane of division. While in the soap bubble, it's the opposite, right? The soap bubble is trying to minimize its energy and get to that configuration because it's a minimum of energy. It's the other way around, if you will, in the plants. Okay? So it's published if you want to see the paper.
What about the fifth shape? Does that correspond to negative curvature?
What shape?
The fifth shape, you gave us the geometry, the angle corresponding to the first four.
Yes.
Have you looked at the fifth one?
So it's not, Etienne is doing the simulations.
Does it correspond to negative
right
curvature?
I don't think it comes out in any of this, even in the plane, it's not an attractor. It is a fixed point, so you can do it, it's right there, but it's not attracting. Or if it's attracting, it will be a very, very small neighborhood around that shape. And we've never seen it. I would love to see it, but we've never seen it.
Okay.
So I was curious about the role of the microtubules, but if we go further, which is how does the cell know that it has accurately divided in half?
So this is, in fact, the more lax part of the division. The cell seems more preoccupied about minimizing the length of finding than the actual sizes of the daughter cells. And we often see differences of up to 20, 30% in the size of the cells. So I think what is going on is when the cell, there's still a lot of biology to be done, but when the cell is about ready to divide, the nucleus takes a central position. So it's brought into the middle of the cytoplasm, also by microtubules and actin. And I think this is essentially how they maintain size. They don't try to measure volume. They just bring a structure, in this case the nucleus, in the center. And when they divide, they know, well, the nucleus will give you the two daughter nuclei. And it's essentially dividing starting from the center of the cell and dividing this way without trying to sum up or do the integral of the volume on either side to make sure that they get 50-50.
So I was curious because in bacteria, there's a reaction diffusion system that finds the center. And I wonder, evolutionarily, if we have any idea where these diverge.
Oh, a long time ago. They don't. No, in terms of division, they have nothing in common. So they don't, for example, they don't have the same microtubules and actin that you see in animal cells and plant cells. they're not seen, or they have other versions, but they're not seen and not nearly as important in bacterial division.
There are exceptions to this rule of dividing in the shortest. You have cells in between the meristem and the primordium in the sort of fold where you have a negative curvature. In these regions, the cells divide along their length, not along their width. So how come?
Good question. I'll leave that to you. It's your paper, after all. So it's true. There are quite striking examples. Some of them, the most striking example is the cambium. The cambium is where you form wood in a trunk. There you have cells that are extremely long, and despite that, they keep dividing along their length. So they have an aspect ratio that can be 10:1, and despite that, they still divide this way. What seems to be the dominant feature of all of these systems is very strong stress anisotropy. So it's possible, and auxin. So they're all systems where a flow of auxin, which is a plant hormone, is known to be quite important, and also to have strong stress anisotropy. So it's possible that there are some external cues that help the cell choose or change its division plane at those locations.
I want to follow up the question Ray just asked, which is about animal cells. I mean, do any animal cells do this sort of thing? Would they do this sort of thing if there wasn't an enormous amount of additional controlling genetic sort of override going on?
Yeah.
You know, is this sort of thing a basic sort of thing that all cells would probably do if they weren't instructed to do a lot of other things instead? I
Very
have
good
no
point.
knowledge.
Yeah. This, so certainly for plants, I need to emphasize it's typical that this type of divisions where you can predict them precisely is typical of regions where you're trying to build a lot of material. So the meristem, or the apex, where you're growing fast, and you're not trying to do anything sophisticated, like making an eye or a special leaf and so on. So it's rapid cell expansion and division when the outcome of the actual cellular pattern doesn't matter. You're just trying to grow and divide. In animal cells, animal cells unfortunately are amorphous, so they're harder to describe in terms of shape, but recent experiments have been done and published where you can plate an animal cell on a patch of integrins or some proteins that will force it to be squared or rectangular, and you see the same thing. So they observe the same behavior. It's a bit more sloppy, but it's still there. They tend to, in this case, they don't have a division plane as such. It's more the spindle that elongates, but the spindle does elongate in such a way that you're dividing the plane. You're using a plane that's shorter overall.
Is there an obvious reason in terms of just ordinary cost why cells that don't want to do anything special would tend to minimize the area? Is it expensive to build cell wall?
I'm not sure if it's very expensive because the cell wall is a product of photosynthesis. So it comes from the sunlight. So it's cellulose, it's a glucose polymer, so they have plenty of cellulose for their use. But it's possible that in fact, it does minimize the amount of material you use for the cell, but it also means, what it means, and probably has more sense in biology, is it keeps the cell as close to a sphere as possible. So it means you don't have cells that have a very long projection that might be hard to maintain, or there might be poor communication because the cells have been distorted over long series of division. So it's a way of growing a tissue. In fact, we were looking at that. So you probably know that if you tessellate the plane and you want to use the least possible edges, you use an hexagonal array. That's the best way of tessellating the plane. If you cannot reach an hexagonal array by growing and dividing, because whenever you divide, you in fact create cells that have 5 sides instead of 6. And you add sides to the neighbors. So you cannot reach this perfect packing on a growing, dividing tissue. So we think it's the next best thing. The way they grow is they grow, and by choosing the shortest possible plane every time they divide, it means they're not hexagonal, but they're as close as possible from being isodiametric, having the same fairly uniform geometry, compact geometry.
Actually, so kind of related to Professor McIntyre's point.
Can't
Hello,
see you. I can't see.
up here.
Okay.
Better be worth it now. So certainly in epithelial tissue, the pattern of cells, their boundary is often very well approximated by the Voronoi map induced by their cell centroids. I was wondering if you saw a similar thing with your meristems.
Well, I would contest that. There are papers that show that the Voronoi map can be close, but in our case, it's not so good. It's not such a good fit with what we observe. But it's the same idea, right? The Voronoi map or space is you're taking all the points that are closest to the centroid, right? Mm-hmm. And so it's a bit the same idea. You're trying to keep the cell as isodiametric as possible. You define them such that you don't have a lot of edges, and you keep them as compact as possible. So I think they tend to be close to each other, but they're not perfectly in sync.